everyone. I'm Zeynep Önsert, and today I'll be presenting to you my research on the redox-modulating effect of Melissa officinalis within the tumor microenvironment. My research specifically focuses on how the molecular compounds in Melissa respond differently depending on the cellular context, helping disrupt cancerous cells while protecting the healthy ones. Before I move on, I would like to talk a little bit more about myself. I'm a rising senior at Ted Adana College, and I'm specifically interested in biochemistry and biotechnology. I've been involved in a range of projects that connect science and sustainability, and these projects range from the production of a lipase-active biofertilizer to bioremediate the contaminated soils to the investigation of oral pathogenic bacteria. I'm also interested in arts and am the starter of the first community in my school, which is the school arts community. I'm also the starter of our bioplastic company that produces biodegradable alternatives to commercial plastic plant pots. Okay, so in 2020, more than 10 million deaths occurred, and 70% of these deaths occurred in low and middle income countries. This data clearly illustrates why alternative treatment, or at least protection strategies, are crucial in the world where treatment is unequal or unapproachable. Okay, so let's start with ROS, or reactive oxygen species. ROS are crucial molecules that are produced in the cell, mostly in the mitochondria, that facilitate a lot of processes in the cell and normally are scavenged via antioxidants that are naturally found in the body. But when in excess, this ROS causes oxidative damage to DNA, lipids, and proteins, which contributes to genetic instability, which is a hallmark of cancer development. In the early stages of tumorigenesis or cancer development, cells with ROS-induced damage escape apoptotic clearance, which would normally lead to cell death. These damaged cells survive and proliferate, accumulating additional mutations over time, which eventually leads to cancer development, as I mentioned. Okay, let's talk about another problem that is triggered by ROS-inflammation loop. Inflammation is a response of the body to tissue injury and is the outcome of pro-inflammatory cytokines. Improper regulation of inflammation production, which is caused by ROS, could adversely affect the production of pro-inflammatory cytokines, leading to inflammation and pyroptosis. Pro-inflammatory cytokines activate signaling pathways that tell even the damaged cells to survive, divide, and resist apoptosis, which is cell death. This means that cells with mutations caused by inflammation are not removed. They keep proliferating, which increases the chance of tumor formation and tumor growth. Okay, let's talk about this pathway on a molecular level now. Our first step is inflammation activation. ROS damages DNA and also activates the inflammasomes, such as NLRP3, which is the most common inflammation type seen in cancerous cases inside the cytoplasm. The inflammasome processes inactive precursors into their active forms. This eventually leads to pyroptosis. If the cell end damage continues, the inflammasome can also trigger pyroptosis, as I mentioned, which is a type of explosive cell death. During the pyroptosis, the cell ruptures, spilling its contents, including damaged DNA, into surrounding tissue. This leads to more ROS production, which creates a self-sustaining inflammatory microenvironment. Let's connect this information to Melissa now. Melissa officinalis, or lemon balm, contains several phenolic compounds such as rosmarinic acid, caffeic acid, and naringin, which contributes to its redox-modulating effects. On this research, we will focus on the ROS modulation of caffeic acid and apigenin. Let's start with caffeic acid now. Caffeic acid can react with 2 superoxide molecules per molecule. One superoxide approaches the catechol hydrogen atom of caffeic acid, and the other one stacks up with the aromatic ring, as you may see in the figure. The ring on the middle is the aromatic ring of the caffeic acid. Let's move on with apigenin now. Apigenin in the previous studies hasn't been studied in detail, and the 3D structuring of apigenin is the originality of this research. Just like caffeic acid, apigenin reacts with 2 superoxide molecules per 1 molecule, and as you may see in the figure, the distance between the superoxide molecule and the aromatic ring of the apigenin is relatively short, which is 2.58, and this means that the reaction is relatively strong, which indicates that apigenin is a really strong superoxide scavenger. Okay, here's a key insight. In tumor cells, Melissa behaves differently than in healthy cells. Its key compounds can act as pro-oxidants under high oxidative conditions. Molecularly, these polyphenols interact with iron in cancer cells. This triggers Fenton-type reactions, which generate highly reactive hydroxyl radicals. These ROS cause mitochondrial dysfunction, which leads to depolarization of mitochondrial membrane potential, which causes release of cytochrome C into cytosol, which triggers activation of caspase-3, which eventually results in programmed cell death. In short, Melissa exploits the already high ROS levels in tumors and pushes them beyond the oxidative threshold, selectively killing cancer cells. To conclude, cancer prevention must be affordable, accessible, and scalable. This research explores how a widely available medicinal plant, like Melissa, may support cancer suppression by targeting mechanisms like oxidative stress and inflammation. We need tools like this. Not expensive therapies limited to a few, but truly accessible and preventive treatment strategies. This may bring us closer to something that matters even more than innovation: health equity. These are the sources that support this work. I would like to specifically thank to Professor Miriam Rossi for her scientific mentorship, especially in the 3D structuring of the molecules. Thank you very much for listening. I hope this research contributes to further investigation on these topics. You may see my contact information on the screen.